Welcome, everyone. Let's jump right into this explainer. Today, we're taking a graduate-level look into the very operating system of life. We are opening up the Molecular Toolkit, Advanced Biotechnology. We're going to trace this absolutely breathtaking scientific arc, starting from simply reading raw DNA sequences all the way to actively rewriting the genetic code itself. It is a wildly exciting time to be studying biology, so let's get right into the molecular mechanics of how we manipulate the fundamental building blocks of life.
Here is our roadmap for today. We'll build our understanding sequentially. 1. Reading the genetic code. 2. Amplification and gene cloning. 3. Analyzing gene expression patterns. 4. Genome editing with CRISPR-Cas9. 5. Cloning in stem cells. And finally, 6. Applications of modern biotechnology.
All right, let's kick things off with Section 1, reading the genetic code. Because, before we can even think about editing the code, we really have to understand how to unravel and read those specific base pairs.
Okay, let's dive into nucleic acid hybridization. This concept is literally the master key that unlocks almost all of our DNA technologies. And the cool part, it relies on a beautifully simple physical property, complementary hydrogen bonding. Adenine binds to thymine, guanine binds to cytosine. By engineering a known single-stranded nucleic acid probe, we can basically fish out its exact complement from a vast genomic ocean. It's elegant, it's highly specific, and as we'll see, it is the absolute engine behind sequencing, PCR, and even CRISPR.
So how do we scale that up? Well, we've completely industrialized the hybridization process with next-generation sequencing. Here's how it works. We take genomic DNA, fragment it, and isolate each piece on its own microscopic bead. After amplifying the fragment so we've got millions of identical copies on that single bead, we do something called sequencing by synthesis. We wash the beads with one type of nucleotide, a dNTP, at a time. When DNA polymerase successfully adds a complementary nucleotide, it releases pyrophosphate, which triggers a tiny, microscopic flash of light. By recording these literal flashes of light across millions of wells simultaneously, we achieve this massive, high-throughput parallel sequencing.
Moving right along to Module 2, Amplification and Gene Cloning. Now we transition from just reading the code to actually isolating and copying specific genes of interest from that massive, unwieldy genome.
To pull this off, we need molecular scissors, formerly known as restriction endonucleases. Evolutionarily, these actually started out as a bacterial defense mechanism to chop up invading viruses. And to protect their own DNA from getting sliced, bacteria just add methyl groups to their adenines or cytosines. Brilliant, right? Well, we've co-opted these highly specific enzymes to cleave DNA at very precise, usually palindromic restriction sites.
This brilliantly illustrates the elegance of molecular cloning. The most useful restriction enzymes make these staggered cuts in the sugar phosphate backbone, leaving overhanging single-stranded segments that we call sticky ends. And thanks to our old friend complementary hydrogen bonding, any two pieces of DNA cut with that exact same enzyme will naturally find each other and temporarily bind. Then, we just drop in DNA ligase to catalyze the covalent phosphodiester bonds, sealing the backbone, and boom, we have a stable recombinant DNA plasmid.
But wait, what if you only have a minuscule, barely there trace of a target gene? Enter PCR, the polymerase chain reaction. The PCR cycle of denaturation, annealing, and extension gives us exponential amplification. But the absolute unsung hero here is Taq polymerase. See, a standard human DNA polymerase would permanently denature at the 95 degrees Celsius heat needed to separate those DNA strands. But Taq polymerase? It comes from Thermus aquaticus, a bacterium that literally lives in boiling hot springs. It thrives in that heat, which allows our automated reaction to churn out billions of copies in just a few hours.
Okay, on to Section 3, Analyzing Gene Expression Patterns. Having the DNA blueprint is fantastic, but we need to figure out when and where these genes actually roll up their sleeves and do their work.
Let me pose a molecular puzzle to you. Let's say we want to use bacteria as microscopic factories to churn out eukaryotic proteins like human insulin. How on earth do we overcome the presence of non-coding introns? Remember, eukaryotic genes are absolutely loaded with them, but bacteria don't have the spliceosome machinery to cut them out of the RNA. If you just shove raw human DNA into a bacterium, it's going to translate a massive, completely non-functional protein. So what's the workaround? Complementary DNA, or cDNA. We basically let the human cell do the heavy lifting of transcribing and splicing the mRNA,
Then we extract that mature, fully spliced mRNA, the one with the poly-A tail. Using a poly-DT primer and a cool little enzyme hijacked from retroviruses called reverse transcriptase, we synthesize a DNA strand backwards, directly from the mRNA. This gives us a perfectly streamlined exon-only DNA sequence that bacterial ribosomes can read without a hitch.
Now, to map expression across entire systems, the field is largely pivoted from DNA microarrays to RNA-seq. The real game changer here is that RNA-seq doesn't restrict you to only looking for sequences you already have probes for. You just grab all the mRNA in a tissue, convert it to cDNA, and sequence the whole lot of it. This gives us this massive dynamic range of expression levels and beautifully highlights alternative splicing variations. It's like getting a high-def, real-time snapshot of the cell's entire transcriptomic state.
Section 4. Genome Editing with CRISPR-Cas9. This is it. We are diving into the most revolutionary tool in our entire kit, the system that finally gives us the power to actively rewrite the genome inside living cells.
Much like restriction enzymes, CRISPR evolved as a bacterial immune system. But instead of cutting its static, hardwired sites, the Cas9 nuclease actually takes instructions. To harness this, scientists engineer a specific guide RNA molecule that acts like a molecular homing beacon. The Cas9 protein binds to it, scans the genome for the exact complementary match, and decisively snips both strands of the target DNA.
But the truly fascinating part is what happens next. That double-strand break essentially panics the cell, triggering its natural repair machinery. If we just let it be, the cell uses non-homologous end joining. It's a sloppy process that randomly inserts or deletes nucleotides, causing a frame shift that permanently knocks out the gene, which is amazing for researching gene function. But for clinical gene therapy, we can step in and provide a normal DNA template. The cell uses homology-directed repair to seamlessly patch the break with our engineered sequence. We are literally correcting a genetic mutation at the source.
Moving into Module 5, Cloning and Stem Cells. We are zooming out from molecular fragments to whole cell reprogramming and organismal cloning.
Somatic cell nuclear transfer. This is the exact technique that gave us Dolly the sheep. We take an unfertilized egg, remove its nucleus, and swap in the diploid nucleus of a fully differentiated adult cell. But here is the massive biological hurdle. An adult skin cell's DNA is heavily modified by epigenetic factors like DNA methylation, which turn off non-skin genes. For the cloned embryo to actually develop, the egg's cytoplasm has to rapidly strip away those methyl groups and reverse the chromatin structure back to a blank embryonic state. Incomplete epigenetic reprogramming is exactly why cloned animals so frequently suffer from developmental defects.
Which brings us perfectly to stem cells. Embryonic stem cells are pluripotent. They can become any tissue, but deriving them from blastocysts comes with heavy ethical controversies. The absolute game changer was Shinya Yamanaka's Nobel Prize-winning discovery of induced pluripotent stem cells, or iPS cells. By using retroviral vectors to drop just 4 master regulatory transcription factors into an adult skin cell, scientists can wipe the epigenetic slate clean. It's basically cellular time travel, returning a differentiated cell back to a pluripotent embryonic-like state.
Let's wrap things up with Section 6, Applications of Modern Biotechnology. We've talked a lot of theory, so let's ground this in its profound real-world impacts.
The clinical triumphs we've achieved with these tools are just staggering. By understanding that chronic myelogenous leukemia is driven by an overactive tyrosine kinase, scientists synthesized Gleevec, a small molecule that perfectly blocks that specific protein, sending patients into complete remission. We are treating severe combined immunodeficiency by using retroviral vectors to insert functional genes right into bone marrow. We've got transgenic goats producing human antithrombin in their milk. And of course, RT-PCR allows us to rapidly detect the RNA genome of HIV long before the patient's body even forms antibodies.
This technology has completely transformed the justice system too. Forensic genetic profiling doesn't bother sequencing the whole genome. It targets short tandem repeats, or STRs. These are highly variable non-coding regions. Just look at this data from the Innocence Project. The PCR amplification of just a couple of STR loci proved definitively that the crime scene DNA, which had 17 and 19 repeats at marker 1, absolutely did not match Earl Washington, who only had 16 and 18 repeats. This exact molecular precision exonerated him after 17 years in prison, while successfully pointing to Kenneth Tinsley.
We're even seeing ecology-scale applications now. By combining CRISPR with gene drives, scientists can engineer a mutation, say making a mosquito unable to carry malaria, and guarantee that mutation is inherited by nearly 100% of offspring instead of the standard 50%. This biased inheritance could rapidly push an engineered allele through an entire wild population. We're talking about the potential to eradicate insect-borne diseases entirely.
However, wielding this kind of power requires immense humility and caution. We are bypassing millions of years of slow, natural selection. Eradicating a species of mosquito could have unforeseen, cascading ecological consequences that we simply cannot predict right now. The power to alter entire ecosystems at the molecular level carries an equally massive burden of responsibility.
Which leaves us with this final, profound question to consider. We now possess the toolkit to edit zygotes, to alter the genome of a human embryo, passing those engineered traits down to every single generation that follows. This is no longer sci-fi. It is biologically possible today. As a society, we are actively navigating exactly when or if we should ever alter human genetics. Where do we draw the line between treating a devastating genetic disease and artificially directing human evolution? It's a complex, heavy landscape, and it's the defining question the next generation of scientists must answer. Thank you so much for joining me for this explainer. Keep questioning, keep learning, and I'll catch you next time.